For 100 years, everyone thought they knew where Irish travelers came from. They were wrong. The story most people accepted without question went like this. Irish travelers were a hybrid population, the descendants of settled Irish families who had, at some point, mixed with Roma gypsies passing through the countryside. It was repeated in newspapers, in casual conversation, in academic footnotes, treated less like a theory and more like settled fact. Nobody had actually tested it against real DNA. That changed in 2017, when a genome study went looking for the truth buried in the actual genetic code of the traveler population, not in folklore passed down for generations. What the researchers found didn't just tweak the old assumption, it inverted it completely, and the fallout reached further than anyone expected. Irish travelers number somewhere between 29,000 and 40,000 people today, roughly 0.6% of Ireland's total population, a group that had remained almost entirely invisible in genetic research until this study came along. Small in number, but carrying, buried in their chromosomes, an entire hidden history nobody had bothered to look for. Over the next 20 minutes, the tour runs through what that DNA actually revealed. The exact generation the split from settled Irish society happened. 4 hidden genetic subgroups nobody knew existed, a disease legacy written directly into the genome, and a political recognition in Dublin that landed within days of the science going public. Is any of it what people expected? Not even close. If genetic mysteries like this are your thing, hit subscribe. There's a lot more buried in the data than the headlines ever covered. To understand exactly how wrong the old theory was, you first have to understand what everyone believed before a single DNA sample was ever sequenced. Because the myth didn't come from nowhere, and untangling where it came from is the only way to appreciate how completely the data tore it apart. For decades, 3 competing theories circulated to explain where Irish travelers actually came from, and none of them agreed with each other. The first was Roma admixture, the idea that traveler ancestry included a meaningful genetic contribution from Roma gypsy populations. The second was famine-driven displacement, the assumption that travelers were simply settled Irish families who lost their land during the devastation of 1845 to 1852 and turned to the road out of desperation. The third, less popular but still present in academic circles, proposed a pre-Gaelic origin entirely separate from the general Irish population. Each theory had defenders, each theory had holes. The famine theory assumed a specific historical trigger, mass eviction and starvation, pushed a whole community into permanent nomadism almost overnight. The Roma theory leaned heavily on cultural and linguistic overlap, assuming that shared vocabulary or shared lifestyle meant shared bloodlines. Linguist Jean-Pierre Liégeois had gone as far as arguing that the traveler dialect known as Gammon carried roughly 10% Romani-derived vocabulary, and for many people, that linguistic detail was treated as proof enough of genetic mixing. It wasn't proof of anything genetic at all. It was proof of contact, borrowing, and centuries of coexistence, nothing more specific than that. What none of these theories had going for them was an actual genetic test, not one. Historians argued with folklorists argued with linguists, and geneticists mostly stayed out of it entirely, because the technology to settle the question with confidence simply hadn't existed until relatively recently. That standoff dragged on for more than 100 years with no resolution in sight, 3 separate stories about the same population, each incompatible with the others, each unverified. In 2017, a team in Dublin decided to stop arguing and start sequencing. That team was led by Edmund Gilbert of the Royal College of Surgeons in Ireland, working alongside Giampiero Cavalieri, also of RKC, along with Shai Carmi of the Hebrew University of Jerusalem, James F. Wilson of the University of Edinburgh's Usher Institute, Sean Ennis, and researchers from University College Dublin. Their findings were published in Scientific Reports, a journal under the Nature banner, in February of 2017. What made this study different from everything that came before it was the resolution of the data itself. Every previous attempt to trace traveler ancestry had relied on blood group markers, a genetic tool so coarse it could barely distinguish broad ancestry, let alone fine population structure. This was the first time anyone had applied a genome-wide, dense SNP dataset to the traveler population specifically. That distinction mattered enormously. Blood group data could hint at a shared origin. It could never resolve the exact timing of a divergence, and it certainly couldn't detect subtle internal structure within a single community. Dense genomic data could do both, provided the team could gather enough of it. The goals were deliberately layered. Three separate unknowns the researchers wanted to settle in a single project. First, ancestral origin. Where did travelers actually come from genetically? Second, internal population structure. Was the traveler community one uniform group, or something more complicated? Third, and perhaps most ambitious, the exact timing of divergence from the settled Irish population, something no one had ever successfully dated before. Solving even one of these questions would have been a meaningful contribution. Solving all three in a single paper was the real ambition behind the project, and it required building the most detailed comparative dataset ever assembled for this population. The first problem the researchers ran into wasn't methodology. It was simpler and more practical than that. The first problem was even finding enough traveler DNA to test. The sample at the center of the study numbered between 42 and 50 individuals who identified as Irish travelers. A small cohort by modern genomic standards, but enough to run meaningful comparisons against much larger reference populations. Set against that core group were 143 European Roma samples, brought in specifically to test the old admixture theory directly rather than relying on assumption. Alongside that came 2,232 settled Irish individuals, forming the primary comparison population against which every traveler genome would be measured. British ancestry was represented by 2,039 individuals, providing a wider Isles-level context beyond Ireland alone. And behind all of that sat a much larger backdrop of nearly 6,000 European and worldwide samples, giving the researchers a global reference frame to rule out coincidental genetic overlap with any other population entirely. The traveler sample size itself, modest as it was, reflected two separate realities. One was simple demographics. Travelers make up a small fraction of Ireland's overall population to begin with. The other was historical and social. Decades of stigmatization and marginalization had left many in the traveler community understandably wary of participating in research conducted by outside institutions. Every one of the 42 to 50 participants who did come forward represented a meaningful act of trust in a process that had rarely served their community well in the past. With those comparison groups locked into place, Roma, settled Irish, British, and the broader European backdrop, the dataset was finally complete enough to start answering the questions the team had set out to resolve. And the first test the researchers ran wasn't a minor or incidental one. It went straight at the assumption everyone already believed they understood. The theory so widely repeated that most people treated it as background fact rather than something requiring proof at all. With the comparison groups locked in, the first question the team asked was the one everyone assumed they already knew the answer to. The first genetic comparison run was traveler against Roma, tested directly against real genomic data rather than assumption or folklore. The result was unambiguous. There was no evidence of recent shared ancestry between the two groups whatsoever. The hybrid population theory, repeated in press coverage, in casual conversation, in academic footnotes for generations, found zero support anywhere in the genome, not a trace. This result directly contradicted assumptions baked into earlier surname studies and cultural analyses, work that had treated linguistic or lifestyle overlap as if it were interchangeable with genetic overlap. It wasn't. Cavalieri stated the conclusion plainly. The Roma descent theory should be considered disproven outright, not simply weakened or left open to interpretation. The distinction mattered. A weakened theory still leaves room for doubt. A disproven one closes the door. And this particular door had been propping open a century-old assumption that most people had simply never thought to question in the first place. The failure of the Roma theory wasn't a quiet non-result, buried in a footnote. It reshaped the entire framing of the paper, because it meant every prior explanation for traveler distinctiveness that leaned on outside admixture now had to be abandoned entirely. The data hadn't just failed to confirm the old story. The data actively pointed in the opposite direction, toward an explanation rooted entirely inside Ireland, rather than any outside population at all. That left the researchers with a genuinely open question, one the old theories had never actually answered, because they'd been too busy arguing about Roma ancestry to consider it properly. If travelers weren't part-Roma, weren't a hybrid population, weren't the product of mixing with any outside group at all, then the obvious next step was to measure exactly how Irish they really were, and how that Irish ancestry compared, genome for genome, to the settled population living right alongside them. Genome-wide comparison placed Irish travelers firmly inside the broader Irish genetic cluster, not floating somewhere outside it as an unrelated population. This result confirmed what decades-old, lower-resolution blood group studies had already hinted at. but had never been able to prove with real statistical confidence. At the broadest level, travelers and settled Irish people share the same ancestral population, the same deep genetic roots stretching back long before either group existed as a distinct social category. That much was clear from the very first pass through the data, but the researchers were careful not to overstate the closeness. Close did not mean identical, and the genome made that distinction obvious almost immediately. Small but statistically significant differences persisted across the traveler genome when measured against the settled Irish reference group. Differences too consistent and too widespread to dismiss as noise or sampling error. These differences couldn't be explained by Roma admixture, because that theory had already been ruled out entirely in the previous comparison. Something else was clearly responsible for the gap, some force operating independently of any outside population mixing with the travelers at all. Whatever it was, it had left a measurable fingerprint across the genome, consistent enough to show up reliably across dozens of samples and thousands of comparison points. The researchers now had two solid conclusions locked in: a shared Irish origin and a real, quantifiable genetic distance from the settled population living alongside them. Neither conclusion made sense on its own without the other. Shared origin explained the similarity. It said nothing about the difference. Figuring out what produced that difference meant turning away from ancestry entirely and looking instead at population dynamics, the kind of slow, structural forces that reshape a genome over generations without any outside population ever being involved. That something else turned out to be one of the oldest and most well-documented forces in all of population genetics, one geneticists have studied in isolated communities across the world for decades. Travelers showed clear features of what geneticists call a genetic isolate, a population that has bred largely within itself for generation after generation, with limited genetic exchange from outside groups. Cavalieri described the resulting genetic distance between travelers and settled Irish people as comparable to the distance separating the Irish from the Spanish. That comparison is worth sitting with. Two populations living on the same island, speaking the same language, sharing recent common ancestry, and yet carrying a genetic gap on par with two entirely separate European nations. The isolation responsible for that gap predates the Great Famine of the 1840s, ruling out what had long been the most popular folk explanation for traveler distinctiveness. If the divergence had started before the famine even happened, the famine simply couldn't be the cause. Two specific mechanisms were identified as the real drivers behind the divergence. The first was genetic drift, the natural, random shifting of gene frequencies that occurs in any population over time, accelerated dramatically in smaller groups. The second was a shrinking population size itself, which intensifies the effects of drift by narrowing the genetic pool available in each generation. Together, these two forces, drift and population size, produced exactly the kind of measurable genetic distance the researchers had already confirmed in the previous stage of the analysis. What the study had effectively done at this point was separate two questions that had been tangled together in public understanding for 100 years: where travelers came from, and when they became genetically distinct from everyone around them. Those are not the same question, and treating them as one had been part of what kept the old theories alive for so long. Origin was now settled, timing remained open. Pinning down that second question, the exact moment the divergence began, required a different kind of genetic clock entirely, one built specifically to measure the passage of generations rather than the direction of ancestry. Researchers turned to multiple independent genetic dating methods to estimate exactly when the divergence between travelers and settled Irish people began, cross-checking each method against the others to avoid relying on any single technique alone. The estimates converged with striking consistency. Across every method applied, the split landed somewhere between 8 and 14 generations back from the present. The best fit estimate settled on 12 generations, translating to roughly 360 years of separation. Counting backward from the study's publication placed that divergence at approximately the year 1657. That single date carried enormous weight. It pushed the emergence of the traveler population as a distinct group up to 200 years before the Great Famine of 1845 to 1852. The very event most people had assumed, without evidence, explained their origin in the first place. If the divergence had already begun 2 centuries before a single potato crop failed in Ireland, the famine simply could not have been the cause, no matter how deeply that assumption had settled into popular memory. Cavalieri was careful about how far to take this conclusion. He emphasized that the study measured social divergence, the point at which the two populations stopped intermarrying and began drifting apart genetically. Not necessarily a single, discrete founding event with a clean beginning. Populations rarely split cleanly on a single day. The number represented a statistical center point, the moment the data suggested traveler and settled Irish genomes stopped mixing in any meaningful way. Not a historical record of one specific incident. Still, the number itself demanded context. 1657 wasn't a random or forgettable year plucked from Irish history. It sat in the middle of a period widely remembered as one of the most violent and disruptive decades the country had ever experienced. And that timing alone was enough to reframe the entire conversation around where this divergence actually came from. The year 1657 falls squarely within the era of Oliver Cromwell's campaigns across Ireland, a period marked by mass land confiscation, forced displacement, and sustained violence against the Irish population. Researchers pointed out that this timing lines up closely with widespread upheaval, entire communities losing land, losing homes, and being pushed into unfamiliar and unstable circumstances almost overnight. A population thrown into that kind of chaos would have every reason to turn away from settled life and toward permanent movement as a means of survival. An alternate theory pushes the possible origin even earlier, back to the late 1500s and the plantation of Ulster, a large-scale land settlement program that displaced existing populations to make room for new landholders. Either scenario, Cromwellian violence or plantation-era displacement, tells fundamentally the same kind of story. Traveler origin as a direct response to catastrophic social upheaval, just not the specific upheaval most people had spent generations assuming. Cavalieri was direct about the limits of what the genetic data could actually prove here. He cautioned that pointing specifically to Cromwell as the cause remained speculation, even though the genetic dating fit neatly inside that historical window. Correlation in timing is not the same as documented causation, and the researchers were careful not to overstate a connection the genome alone couldn't fully confirm. What the data could say with confidence was this: the famine myth wasn't just wrong. It was wrong by roughly 2 centuries, and the actual period of displacement that likely shaped traveler origins may have unfolded during one of the most brutal chapters in Irish history, rather than the one popular memory had settled on. But timing was only half the puzzle the genome had to offer. The same dataset that dated the divergence also revealed something else entirely, something that complicated the picture even further. The traveler community, it turned out, wasn't one single, uniform group at all. Fine-scale genetic analysis, run beyond the broad comparisons already completed, revealed 4 distinct genetic clusters existing within the traveler population itself. This internal structure had never been documented in any previous study, genetic or otherwise. Nobody had gone looking for it before, and without the resolution of dense genome-wide data, nobody could have found it even if they had tried. Crucially, these 4 clusters weren't scattered at random across the sample. They mapped directly onto real-world social and linguistic groupings already recognized within the traveler community itself, patterns of marriage and association that had shaped the population from the inside for generations. In practical terms, this meant the label Irish traveler, treated for 100 years as a single genetic category, was technically an oversimplification. 4 related but genuinely distinguishable founder groups existed underneath that single umbrella term, each carrying its own internal history of marriage patterns and social boundaries. The discovery reframed the entire population, no longer a single homogeneous group, but a mosaic, 4 separate threads woven together under one shared cultural identity. That reframing opened an entirely new set of questions the original study hadn't set out to answer. How had each of these 4 clusters formed independently in the first place? What social or geographic boundaries kept them distinguishable from one another across so many generations? Had these clusters always existed since the initial divergence from the settled Irish population, or had they emerged later, splitting further apart from an already isolated group over time? The data could confirm the clusters existed. It could not yet fully explain how each one came to be. One of those 4 clusters, though, carried a name that wasn't unfamiliar or obscure to anyone inside the community at all. One genetic cluster aligned specifically with what researchers called the Rathkeil group of travelers, a name drawn directly from the County Limerick town long associated with this particular branch of the community. Rathkeil itself has historically stood as one of the most prominent and economically significant traveler communities anywhere in Ireland, a name recognized well beyond genetic circles long before this study ever existed. What the genome data added was something new. statistical confirmation that this group had maintained relatively distinct marriage patterns compared to other traveler branches. Patterns consistent enough to leave a measurable signature in the DNA of its members. This is precisely the kind of fine-grained internal structure that older blood group studies from previous decades were never sensitive enough to detect. Those earlier methods could tell researchers, in broad strokes, whether two populations were related at all. They had no ability to resolve differences this subtle, differences that required dense, genome-wide sequencing to even register as statistically meaningful. So for generations, the distinctiveness of the Rathkeil group existed only as lived experience and community knowledge, understood clearly by travelers themselves but never confirmed by outside science. The 2017 study changed that. It took something long understood only anecdotally within the community and gave it genetic weight. Hard data sitting alongside the oral and social history that had always pointed to the same conclusion. That confirmation mattered, beyond simple curiosity. It demonstrated that the internal boundaries travelers themselves recognized as real and meaningful were not just social constructs layered on top of a genetically uniform population. They correlated with something measurable at the level of the genome itself. Rathkeil represented one clear example of that correlation. A single named branch whose distinctiveness now had two forms of evidence behind it: community memory and DNA. But Rathkeil was only one of 4 clusters identified in the analysis, and the origin of the other 3 didn't trace back to a specific town or region at all. The other 3 clusters split along a different line entirely: language. 2 of the remaining 3 genetic clusters correlated directly with which dialect of Shelta a traveler's family spoke, dividing along the line between Cant and Gammon rather than geography. Shelta itself is the traditional language of Irish travelers, built from a mixture of Irish, English, and invented vocabulary developed and maintained within the community over generations. What makes this particular finding stand out is the fact that dialect and genetics lined up so consistently across the sample. That correlation suggests something specific. Language use and marriage patterns weren't operating independently of each other. They reinforced one another over time. Families who spoke one dialect tending to marry within that same linguistic group, generation after generation, gradually producing a genetic boundary that mirrored the linguistic one. Cases where a spoken dialect boundary and a genetic boundary in the same ethnic group actually match this cleanly are rare in population genetics. Most linguistic divisions blur across genetic lines within a generation or two, diluted by intermarriage that ignores language entirely. Here, that dilution didn't happen, at least not enough to erase the pattern from the genome. The implication is direct. These traveler subgroups functioned almost like semi-separate breeding populations for centuries, distinguishable not just by which words they used at home, but by measurable genetic markers passed down through generations of relatively closed marriage networks. Taken together with the Rathkeil finding, this pattern strengthens the broader case running through the entire study: that Irish traveler identity contains real, measurable internal diversity, not a single monolithic genetic profile hiding under one cultural label. 4 clusters, 2 organizing principles, 1 population. That same internal closeness, the practice of marrying within small defined groups generation after generation, came with a genetic cost the researchers had to quantify next. Researchers turned next to a specific metric: autozygosity, a measure of the degree to which a person's 2 gene copies are identical because they descend from a shared ancestor somewhere further back in the family tree. The results were striking on their own terms. Traveler autozygosity levels came back comparable to those found in Orcadian populations, descended from first or second cousin unions. The Orcadians, from Scotland's Orkney Islands, rank among the most studied inbred founder populations anywhere in European genetics, a group geneticists have used for decades as a benchmark for measuring the effects of close relative marriage on a population's genome. Placing traveler autozygosity in the same range as that benchmark population was not a small statement. Consanguineous marriage, cousins marrying cousins, has historically been common within traveler communities, a fact long known anecdotally and discussed within the community itself. What this study did was take that cultural reality and translate it into hard genomic data, measurable at population scale, rather than treat it as folklore or assumption. It wasn't random cultural trivia dressed up as science. It was a documented, quantifiable feature of the genome itself, visible in the same dense SNP data used throughout the rest of the analysis. Isolation and consanguinity together didn't operate as separate forces. They amplified each other, compounding the effects of the genetic drift already documented earlier in the study, and pushing autozygosity levels higher than they would have reached through isolation alone. A community that most outside observers had long assumed was simply poor and marginalized, nothing more complicated than that, turned out to carry a level of genetic isolation on par with one of the most clinically studied founder populations in all of Europe. That level of isolation doesn't stay abstract. It doesn't remain a statistic confined to academic papers and comparison charts. It shows up in real, diagnosable disease, carried forward generation after generation inside the population itself. Higher rates of specific recessive genetic disorders have been documented directly within the traveler population, and researchers link them squarely to the consanguinity and isolation measured in the previous stage of the analysis. Transferase-deficient galactosemia occurs at elevated rates compared to the settled Irish population, a metabolic disorder tied to a recessive gene variant that becomes far more likely to surface when both parents carry the same rare mutation. Hurler syndrome follows the same pattern. Also known as mucopolysaccharidosis type 1, caused by mutations in the ADUA gene, it appears with noticeably higher frequency among travelers than in the general Irish population. Eye cell disease has also been catalogued among the inherited disorders specifically documented within this community, adding a third named condition to the disease profile researchers assembled. What stands out across all three of these conditions is where the underlying mutations actually come from. Many of the causal genetic variants match those already found in the settled Irish population. The mutations themselves are Irish in origin, nothing foreign or newly introduced. What changed was concentration, generations of intermarriage within a smaller and more isolated population, taking rare recessive variants that exist quietly and harmlessly in the wider Irish gene pool and pushing them together far more often than chance alone would predict. That's the direct, measurable consequence of the autozygosity documented in the previous stage of the study, translated from an abstract genomic statistic into real clinical outcomes affecting real families. In response to these findings, researchers have called for dedicated genetic counseling and disease mapping resources built specifically for the traveler community. Resources tailored to a population whose disease risk profile differs meaningfully from the general Irish population it otherwise closely resembles. The same week these genetic findings went public, something entirely unrelated, on the surface at least, was happening in the Irish Parliament. On March 1, 2017, just weeks after the genetic studies publication, Taoiseach Enda Kenny stood in the Dáil and formally recognized Irish travelers as a distinct ethnic minority. The announcement arrived after more than 25 years of sustained campaigning by traveler advocacy organizations. Decades of pressure finally reaching a formal resolution. Kenny called it a historic day for the traveler community and a proud day for Ireland. A statement met with a standing ovation inside the chamber itself. From the public gallery, activist Bernard Sweeney called out for something further still, urging the Taoiseach to offer a formal state apology alongside the recognition being granted. The genetic evidence, published only weeks earlier, describing a genuinely distinct and genetically isolated population, lent unexpected scientific weight to a political and cultural argument that had been building for a quarter of a century without that kind of backing. Officials were careful with the framing here. They stated explicitly that the recognition conferred no new legal rights on the community. Its significance was symbolic and identity-based, a formal acknowledgement of distinctiveness rather than a change in law. Science and politics collided almost by accident in that narrow window of time. A peer-reviewed genome paper landing right as a parliamentary process decades in the making reached its own climax. Two entirely separate tracks of evidence arriving at the same conclusion within weeks of one another. The genetic case made for biological distinctiveness sat alongside a cultural and linguistic case that had existed for far longer, built not on DNA at all, but on language. The language argument for traveler distinctiveness turned out to have its own supporting evidence, dating back centuries before any DNA test existed. Linguists Kuno Meyer and John Sampson, working independently of one another, dated the existence of the Shelta language back as far as the 13th century. That timeline places Shelta's origins roughly 300 years before the first Roma populations are believed to have arrived anywhere in the British Isles. The implication is direct. If Shelta already existed as a distinct language before Roma contact with Ireland was even geographically possible, then a major pillar of the old Roma descent theory collapses on linguistic grounds alone, independent of anything the genetic data had already shown. This lines up precisely with the earlier genetic finding of no recent Roma ancestry within the traveler population, two entirely separate fields of research, working with entirely separate forms of evidence, arriving at the same conclusion from different directions. Shelta itself was constructed through deliberate techniques applied directly to Irish, methods like word reversal and consonant substitution, transforming existing Irish vocabulary into something distinct and internally coherent. That construction method is evidence of purposeful, internally generated language change, developed from within the community itself rather than absorbed through mixing with an outside population. The correspondence between the linguistic timeline and the genetic timeline stands as one of the strongest cross-disciplinary confirmations found anywhere in this entire case. Two fields that rarely intersect, population genomics and historical linguistics, reach the same destination from completely different starting points, each ruling out the same discredited theory using entirely different kinds of evidence. Ireland, though, wasn't the only place this exact genetic pattern would eventually turn up. A 2026 study published in Nature Communications examined the genetic ancestry of the Scottish traveler population, using a community-driven research model built in direct collaboration with traveler participants themselves. The results echoed the Irish findings closely. Scottish travelers were found to be genetically distinct from both Romani populations and the settled British majority surrounding them, ruling out the same kind of outside admixture theory that had once been assumed for their Irish counterparts. Their ancestry traced back to Scottish origins specifically, mirroring almost exactly the pattern the 2017 Irish study had already established for its own population. The Scottish research went further still, identifying elevated risk for certain inherited diseases tied directly to founder effects within the community, a disease pattern that echoed the galactosemia and Hurler syndrome findings already documented among Irish travelers. To measure isolation and consanguinity, the Scottish researchers used runs of homozygosity analysis, the very same core genetic technique applied in the original 2017 Irish study, nearly a decade earlier. Two separate national traveler populations, studied roughly a decade apart, using overlapping methodology, produced strikingly parallel genetic stories. The Irish findings, in other words, weren't some isolated statistical anomaly confined to one country and one dataset. An almost identical genetic signature turned up in a completely separate nation's traveler population, distinct in geography and history but strikingly similar in genomic structure. The Irish story didn't stay contained to Ireland or to Scotland either. It crossed an ocean generations ago. An estimated 10,000 descendants of Irish travelers currently live in the United States, scattered across several distinct communities that trace their roots directly back to Ireland. The largest concentration sits in Murphy Village, South Carolina, founded in the 1960s around Father Joseph Murphy, a community that in some accounts traces back to as few as 11 core surnames shared across its population. Smaller communities exist as well, spread across Texas, Georgia, Mississippi, and Tennessee, many of them named for the specific region their ancestors first settled after arriving in the country. Traces of shelta-derived cant persist among travelers in the United States, though generations of sustained contact with American English have reshaped what was once a distinct language into something closer to a regional dialect layered over English itself. Genealogical research has traced traveler family presence in America back as far as the 1760s, a date that predates even the famine-era emigration waves most people assume account for their arrival entirely. That timeline alone undercuts the assumption that Irish traveler migration to America followed the same pattern and the same century as the broader wave of Irish emigration driven by the famine. Cultural practices carried across the Atlantic held remarkably intact across generations as well. Early marriage, arranged matches between families, and dowries exchanged as part of those arrangements, all persisting in American traveler communities much as they had in Ireland itself. Every branch of this story, Irish, Scottish, American, keeps circling back to the same unresolved question. No single founding event has ever been definitively proven for the Irish traveler population. The 2017 study measured social divergence, the statistical point at which two populations stopped intermarrying and began drifting apart genetically, not a discrete origin moment with a clean, documented beginning. Researchers have explicitly called for larger sample sizes going forward, data that could refine both the exact divergence date and the boundaries separating the four internal clusters identified in the original analysis. The precise driver behind the timing, whether plantation of Ulster displacement in the late 1500s or Cromwellian-era violence in the 1650s, remains unresolved, and by the lead researcher's own admission, still speculative rather than confirmed. Disease mapping within the traveler community also remains incomplete. Further funding has been requested specifically to expand that work, building a fuller picture of exactly which recessive conditions carry elevated risk and at what frequency. Whether additional undiscovered subclusters exist beyond the 4 already identified remains an entirely open question, one the current dataset simply isn't large enough to answer with confidence either way. The Scottish parallel raises a further question of its own, one that extends well beyond Ireland and Scotland combined. How many other so-called outsider populations scattered across Europe carry this same hidden genetic signature, unstudied and unrecognized, simply because nobody has ever pointed a genome-wide dataset in their direction? For a group defined for centuries almost entirely by what outsiders assumed about them, the actual data, once someone finally bothered to collect it, told a very different story than the one that had been repeated for generations. The Roma descent myth, repeated for generations as settled fact, was disproven outright by genome-wide data showing no recent shared ancestry at all. The famine origin myth met the same fate, dismantled by genetic dating that placed the actual divergence centuries earlier than the story most people had always assumed. The idea of a single, uniform traveler population gave way to something more complicated. 4 measurable internal clusters, each with its own distinct marriage patterns and, in some cases, its own dialect. And the assumption that any of this was uniquely Irish collapsed as well, echoed almost exactly by a separate 2026 study of the Scottish traveler population, built using the same genetic tools nearly a decade later. The community at the center of all of it received formal ethnic recognition within weeks of the genetic case being made public. 2 entirely different forms of evidence, political and genomic, converging on the same conclusion within the same narrow window of time. If one small, overlooked population held this much hidden genetic history buried in its DNA all along, the real question left standing is how many other communities out there are still waiting for someone to actually look.